Good evening, students. Last class we have completed a fission method. There are two type of fission method we have completed last class. One is binary fission, another one is multiple fission. And today I am going to teach about the budding. So budding, nothing but it is a process of formation of small bud or small elevation from the parent body. So there are two kinds of budding methods seen in organism. One is exogenous. That means the bud develops from the external surface of the body or from the outer surface of the body. And second one is the internal budding or endogenous budding. Here the bud develops into the inside of the parent's body. Let's see one by one. And this is a first example for a exogenous budding. This exogenous budding seen in Leucosolinia. Leucosolinia nothing but a sponges. Leucosolinia. In this technique, the parent body produces one or more bud. Here see the picture, one or more bud may develops from the parent body, and each buds grow into the young ones. The bud separated from the parent body and leads to a normal life. So this considered as a daughter individual or this young ones or young bud. This young bud, after the completion of their growth, they detached from the parent body and will grow into the new organism. In sponges, that means in Leucosolinia. This name is given in your textbook. Leucosolinia. The buds constrict and detached from the parent body, and the bud develops into the new sponges. So this is a first kind of exogenous budding seen in sponges, Leucosolinia. It is a type of marine sponges, Leucosolinia. That's all. Next we move on to the another type of exogenous budding. This type of budding seen in Hydra. Hydra is a diploplastic animal. That means the body wall made up of two layers, namely ectodome and endodome. And inside the body, the cavity is there. The name of the cavity is gastrovascular cavity. So these are the three characteristic features of Hydra. First one is ectodome, then endodome, and third one is gastrovascular cavity. When food is plenty, when there is availability of plenty of food, the ectodome cells increase and to form a small elevation on the body surface. So this type of bud formed during when there is availability of plenty of food. So during that time, the ectodome increases. So this ectodome increases, and the endodome also pushed out from the bud. So this is a first step in the budding method. The ectodome and the endodome pushed out from the bud, and this bud contain a interior lumen as it like a parent body. The interior lumen nothing but a gastrovascular cavity, and the bud enlarges and develops a mouth and a circle of tentacles. It's free end. See here, this bud is developed from the parent body, and the tentacles also encircle the mouth. When a fully grown, when it is complete the full growth, the bud constrict and detached from the parent body and will grow into the new organism or new Hydra. So this is a two method or two kinds of budding seen in organism. First kind of exogenous budding seen in Leucosolinia, and second type of exogenous budding seen in Hydra. In both method, the small elevation is formed from the parent body. After complete their full growth, the new buds detached from the parent body and will grow into the new organisms. Next we move on to the endogenous budding. This endogenous budding seen in some dinoflagellate like a Nactiluca. This is a Nactiluca. an example for dinoflagellate. In Actiluca, hundreds of buds are formed inside the cytoplasm. This is a cytoplasm. Here more than hundreds of buds formed from inside the cytoplasm may remain within the body of the parent. This is called endogenous budding. So in this technique, the buds are formed inside the body. That is the reason this kind of budding called as a endogenous budding. This endogenous budding seen in Noctiluca. Next, move on to the next example for endogenous budding. In this kind of endogenous budding seen in sponges, some freshwater sponges and some marine sponges, a regular peculiar mode of asexual reproduction occurs by the internal buds or a gemmule formation. The name of the formation of internal buds are called gemmule formation. In this part, archaeocytes will develop inside the body. Archaeocytes means a complete grown gemmule is hard ball consisting of internal mass of food laden. This part contains a more amount of food, and this structure is called archaeocytes. During the unfavorable condition, these sponges disintegrates, but the gemmules can withstand adverse condition. These gemmules are archaeocytes withstand during the unfavorable condition, but the sponges disintegrates. During the favorable condition, the gemmules are catched and begins into the new sponges. So this is also another example for internal budding. The name of the technique is called gemmule formation. This gemmule formation or the archaeocyte formation occurs during the unfavorable condition. And during the unfavorable condition, what will happen? The gemmules are formed, but the sponges are disintegrates. During the unfavorable condition, when the condition becomes favorable, these gemmules are begins to catch and formed into the new sponges. That's all. Budding method over. So budding, nothing but it is a process of formation of small alleviation from the parent body, or small outer growth formed from the parent body. There are two kinds of budding. One is exogenous budding, another one is endogenous budding. Exogenous budding normally seen in hydra and leucosolinia sponges, and endogenous budding seen in Noctiluca and sponges. So next we move on to another kind of asexual reproduction called fragmentation. Fragmentation, nothing but here the parent's body breaks into smaller fragments or smaller pieces. Every pieces will grows into a new individual. This is called fragmentation. This fragmentation normally seen in some lower organisms. Here we are going to see the fragmentation or pedal laceration in sea anemone. Here a small lobe is constrict in the parent body, and this pedal disc detached from the parent body and will grow into the new sea anemone. So here the fragmented part will grow into the whole organism. That is the reason this process is called fragmentation. Example, sea anemone. So fragmentation may be a 2-mark question. The parent body breaks into smaller fragments or more fragments. Every fragments will grow into the new organism. The type of fragmentation seen in sea anemone is called petal laceration. The another type of fragmentation is called apolysis. This apolysis process seen in Taenia solium. In Taenia solium, this is a segment or a proclatids. This is called matured proclatids or a gravid proclatids. Gravid proclatids may contains both egg and sperm. It is a hermaphrodite organism. This individual can produce both male and female gamete, and these gravid procladids are regularly cut off either single or group to form the posterior end by the process called apolysis. Okay, this is a matured procladids. This matured procladids may detached from the parent body either single or a group by regular interval. This process is called apolysis. So apolysis is nothing but it is a process detachment of some matured procladids from the parent body of Taenia solium. What is the significance of this apolysis process? This apolysis, it is significant in the helping transferring the developed embryo from the primary host man to the secondary host pig. Because of this is a digenic parasite. Digenic parasite means complete their life cycle into two individuals. The primary host man, because of the sexual reproduction takes place in man, and asexual type of reproduction seen in secondary host called a pig. So what is meant by apolysis? Apolysis, it is a process of detachment of gravid procladids or matured procladids from the part of Taenia solium or end part of the Taenia solium. Apolysis over. The last process of asexual reproduction is regeneration. Okay, regeneration nothing but it is a regrowth of last part or injured region. This regeneration was first studied in Hydra by Abraham Tremblay. Abraham Tremblay, the scientist, was studied the regeneration in Hydra. So there are two type of regeneration seen in organism. One is morpholaxis, another one is epimorphosis. What is meant by morpholaxis? Morpholaxis is nothing but it is a process of formation of whole body from the small fragments. A complete or entire organisms will grows into the small fragment or small piece of the parent body. This is called morpholaxis. Example, Hydra and Planaria. This kind of morpholaxis type of regeneration seen in Hydra and Planaria. When Hydra is accidentally cut into several pieces, each pieces can regenerate on the last part and develops into the whole individual. This is called a regeneration or morphologic type of regeneration. What is my epimorphosis? The epimorphosis is nothing but it is a process of replacement of last body parts. Replacement of last body parts. There are two kinds of epimorphosis seen in organism. One is reparative and restorative. Only a certain damaged tissue can regenerate. This is called reparative, and regenerative, the last part can develop. Reparative means the damaged or a certain damaged tissue can regenerate. But in restorative method, the last part, the complete last part can develop. This is called a restorative. Next class we will discuss the sexual mode of reproduction. Thank you.